In my previous video, I made slides based on the basis of the specimen and stained them to see it under the microscope. One of them, which was a prokaryotic cell called bacteria, I wasn't able to give out the proper details on how it's done. On the basis of microbiology, it's commonly grown on a cultural medium and isolated to apply culture sensitivity and its reaction to antibiotics. So in today's video, I will talk about microbiology specifically for bacteria. In nature, common microorganisms like bacteria, viruses, and fungi are commonly discussed to cause diseases and infections to the human body. Both pros and cons are counted to understand the nature of these microorganisms, as well as the study of microorganisms is called microbiology. Viruses are even smaller than bacteria, and they are an infectious agent that exclusively lives inside living cells of a host organism. Because they cannot produce energy or reproduce on their own, so viruses are generally not considered alive. Bacteria are microscopic single-celled organisms that exist all around us and even inside our bodies. Some of them are good bacteria, which is used in a wide variety of applications, while some is bad bacteria known to cause infections. Fungi, however, are a diverse group of living organisms including mushrooms, mold, and yeasts that belong to their own biological kingdom. They are heterotrophs, meaning that they can't make their own food, so they have to absorb nutrients from other surroundings, primarily acting as decomposers. Specifically talking about bacteria, there are two main classes of bacteria, which is a gram-negative and gram-positive bacteria. Gram-negative bacteria are mostly rod-shaped structures which give negative result upon a gram stain test, giving a red or pink color upon staining. Gram-positive bacteria, however, are mostly round spherical shaped structures which gives positive result upon a gram stain test, giving a purple or blue color upon staining. To do this, I mean for growing bacteria, a cultural medium like agar is used to grow this stuff at room temperatures. On the basis of their growth, some common examples of bacteria in terms of gram-negative are like E. coli, Pseudomonas, and Klebsiella. In terms of gram-positive are like Staphylococcus, Streptococcus, etc. To prepare the agar solution, first I had to do a few calculations to prepare my agar base. I wasn't making a lot, so I thought that about 62.5 mils would be just fine and enough. So I got out my scale and measured about 2.5 grams of agar and transferred it into a flask. Then I added about 62.5 mils of distilled water, covered it with foil and mixed it up as best as I could. Then to another flask I added about 3.2 grams of agar along with another 62.5 mils of distilled water, covered with foil and mixed it up as well. Now all I had to do is to incubate and sterilize at 121 degrees C for 15 minutes, so I kept the flasks on my hot plate. After keeping the flasks on my hot plate, I set the temperature to 121 degrees C. At first nothing much happened, but the one on the left started boiling. However, one on the right is not looking good because the flask is not properly flat, so I had to swap it out with another one. This time the one on the left was up to 121 degrees C, so I started my timer for 15 minutes, while the other flask was still warming up. Once the boiling was completed for the smaller flask, I removed it from the heat and let it cool down, while the bigger flask was set to boiling at 121 degrees C. Now in the meantime, I took the flask and removed the foil and let it cool down, and poured the agar solution into sterile petri dishes. Then the same procedure was applied to the other flask. I let it cool down and poured the agar solution into the rest of the petri dishes. Make sure not to solidify the agar in the flask while it's still warm and liquid in it. After 15 to 20 minutes, I let the agar solidify in the plate and let it cool down, and then now it's ready for storage or streaking. For this purpose, the sample can be anything, either be the sample of urine, stool, pus, or any other bodily fluids. For negative control, I applied my own urine sample. First, I sterilized the loop with some heat and then let it cool down. After that, dip it into the sample and streak it onto a plate. Then again sterilize the loop and redo the procedure for another spot of another sample at another area on a plate. For positive control, I had already pre-grown some bacteria and mixed it with some distilled water inside a tube, after which I let it sterilize to be applied on another plate. I streaked onto the two plates of different media and then again let it sterilize over some heat and let it cool down. Now, as for just a small test, I took a small sample of dirty water from a pond filled with water covered in a small spot and applied it onto the plate. Now, the procedure of inoculation is completed and it is required to be incubated at 37 degrees C for 24 hours. So I turned on my incubator and right now it is set to be at room temperature. So I kept my plates inside and let it incubate overnight. The next day I took out my agar plates and it seems that something actually grew. Upon close inspection, the pink color shows the growth of colonies of a common gram-negative bacteria, mainly E. coli, which is the positive control plate that I applied. My own urine sample shows negative result upon inspection, which is good because growth of bacteria can resemble UTIs, urinary tract infection, which is not good. The one which is the experimental test of pond water also shows growth of pink colonies, mainly E. coli. So since I don't have any urinary tract infection, out of curiosity I applied sensitivity of antibiotics to the positive control plate. Antibiotics are powerful medications that treat bacterial infections by killing or preventing bacteria from reproducing. They can either be bacteriostatic or bactericidal, which works either way to suit their own type of bacterial infection, and sensitivity to antibiotics. Most of the time, no proper awareness to people can lead them to improper dosage or use of antibiotics for common bacterial infections, leading them to be either making the bacteria getting resistant to antibiotics. Therefore, proper testing of bacterial infection is required to let the doctors and technicians guide which medication will be better for treating the certain type of bacterial infection. So here are the antibiotics that I am going to use in this process. To do this, I took a small colony of bacteria and applied it on a plate like this. Once it was done, I placed the antibiotics onto the plate using tiny forceps. This way we can apply a dozen of antibiotics rather than giving and finding out which medication would seem better on the patient, which can also lead to drug dependence or addiction to drugs. After this, I placed the whole plate inside the incubator for 24 hours. Sometimes, not knowing the type of bacterial growth due to non-lactose fermentation or NLF, we can apply certain biochemical tests or do a gram staining in order to find out the growth of a specific bacteria. To do this, I took a clean glass slide and sterilized my loop, after which I let it cool down. Then I took a small colony of the bacteria and applied it onto the slide. Then let it fix, overheat and sterilize it. At high temperatures, the bacteria cannot survive. In this case, it just burns instantly and dies in form of carbon, leaving us with a slide to stain. Following stains were added and washed regularly over the course of some time. Then I placed it under the microscope. The pink or red color shows and confirms gram-negative E. coli, which are these rod-shaped structures over here. However, for the gram-positive bacteria comes down to the cell wall of bacteria, which is made up of murine. Gram-positive bacteria like Staphylococcus have a thick cell wall which oftenly adapts to the purple color of crystal violet stain, avoiding the saffronin stain, giving it a nice blue or purple color. However, the gram-negative bacteria like E. coli have a thin cell wall which adapts to both crystal violet and saffronin, which loses the crystal violet after applying the decolorizer and accepts saffronin as the final stain, which gives it a nice red or pink color. After 24 hours, I checked the plate and only one drug remained sensitive. while the others remained resistant, meaning that some other antibiotics may be used to find the better treatment to treat specifically this type of bacteria, giving a much better, bigger zone upon sensitivity. Well, this was all about bacteria in general, and in future I might work on bioluminescent glowing bacteria. I don't know when or where I will get the chance to do it, but I might work on it. But right now I guess I might just work on other types of bacterial growing species. If you like watching videos like these, consider subscribing and liking my videos, and I will hopefully see you in the next one. Thanks for watching.